All right, picking up from our stretch break, we'll continue on into organelles that build proteins. So we mentioned the idea that cells were factories. So it's a really good analogy. Factories build all kinds of things, but cells primarily start with building proteins, and then they use those proteins to build other sorts of things. Um, so when we look at kind of what's going on, we've got ribosomes, which are the actual molecules that do the building. We've got something called endoplasmic reticulum, where we build lipid components of the cell membrane, along with other sorts of things. And then we've got the Golgi apparatus, which kind of acts as the customization shop or packaging center where it sorts, modifies, and gets things ready to head out of the cell. So here's kind of a visual. So ribosomes come in, and we'll talk about this pretty extensively when we get into genetics here. But ribosomes come in two parts. You can think of them kind of like two hands, and they clamp together over this molecule called RNA, which is the part that we're going to read, and we're going to turn that into a protein. And you can see these little protein strands kind of being built off the side here. And then these ribosomes can attach to something called the endoplasmic reticulum, or the ER, and you can see them on here. There's kind of all these little dots here. And what they do is, again, they build that protein, but in this case, they're going to push the protein into the middle space, the inner space of the endoplasmic reticulum. And the reason they can do that, so here we've got kind of two different versions, rough ER and smooth ER. The big difference is the rough ER has the ribosomes on the outside of it. You can see them there, whereas the smooth ER does not. So we're going to make different things, like factories make things. We're going to make different things, and some of them will get pushed into this space, and then it gets kind of funneled through this space, out through these tubes of the smooth endoplasmic reticulum to whatever part of the cell needs those different things there. Some of those will eventually come off as little vesicles and move their way to what we call the Golgi apparatus, which is often described as looking like a stack of flattened pancakes, where they get processed and then they'll get ready to leave the cell to go into other cells to do whatever they need to do there. So 13, what are ribosomes? What do they do? The small parts of RNA and proteins found throughout the cytoplasm involved in protein synthesis. Which organelles the lipid components assembled at, and that's the endoplasmic reticulum. And then what's the difference between rough ER and smooth ER? It's whether or not it has ribosomes attached. And then rough ER has the ribosomes, smooth ER does not. Using the cell as a factory analogy described the role of the Golgi apparatus. So it's, like I said, the customization shop or the postal service. It kind of figures out what things need to be done to the things before they can leave and to go elsewhere there. And then suppose the cell's Golgi apparatus does not function properly. How might this affect other cells? So imagine if you've got a shipping center that sends things to the wrong address or sends out the wrong things. Whatever gets those things or doesn't get those things is going to be in trouble. So we need to make sure that the Golgi apparatus works the same way. Otherwise, the receiving cell will not function appropriately. And then let's talk about cellular energy. So there's two kind of big things we talk about with energy. One are the chloroplasts, and the other are the mitochondria. And we'll look at kind of how these come together here. So visually, chloroplasts have this outer membrane and inner membrane, and then a bunch of membranes kind of all stacked together. They're called thylakoids. And here's a visual of that. You can see kind of that, hard to tell, but there's two membranes on the outer part there, and then there's all this stack of stuff in between. Mitochondria, which we often talk about as the powerhouse of the cell, likewise has an outer membrane and an inner membrane. And the inner membrane is really, really highly folded. And so one of the questions we can ask is, why are these, both of these have so many folds or so much surface area on this inner membrane? And the short answer is that the more surface area you have, the better you are at making energy. So in this case, we're converting solar energy to chemical energy. And in this case, we're converting one form of chemical energy to another form of chemical energy. So photosynthesis captures it, and mitochondria make it available to the cell. So looking at the Venn diagram, chloroplasts found in plant cells and algae cells, they capture energy. They use a molecule called chlorophyll, and they do this with a process called photosynthesis. Mitochondria are found in all eukaryotic cells, that includes plant cells. They release energy to a usable form, and they use it in a process called cellular respiration. And as we mentioned, they both have multiple membranes on there. And the other thing that's really interesting about this is they each have their own set of genetic information. And we suspect that this is because they're probably descended from independent microorganisms, bacteria, that moved into cells a long time ago and just took up residence inside those cells. So filling in our questions here, chloroplasts are never found in animal cells. That is true. Mostly, there is a story of this particular type of sea slugs that can actually eat algae and then use the chloroplast from the algae, and rather than digesting it, they move it to their skin. So the sea slugs can actually have chloroplast, but they're not born with them. They actually have to kind of pirate it from the food that they eat. 20. Unlike chloroplasts, mitochondria are surrounded by a double membrane. That's false. So like chloroplasts, that's true, we have. 21, nearly all the mitochondria in your cell are inherited from your mother, and that is true. And then 22, both chloroplasts and mitochondria lack genetic information in the form of DNA, and that's false. It should be contained genetic information in the form of DNA. All right. And then last little bit here, cellular boundaries. So all cells are, actually we'll pause here. And we'll come back to this for our next part. So I'm going to stop that.